welcome everyone to today's breakfast event. We're very, very pleased to have three very distinguished guests and speakers who will talk to us about Hidradenitis Suppurativa, some of the challenges in research in this very important disease state in dermatology. My name is Jason Shuris. I'm the Chief Business Officer at Innovaderm Research. Next slide, please, Mikhail. Dr. Bedside is our founder and Chief Executive Officer of Innovaderm Research. I'm sure many of you in the room have heard of him or know of him or know him personally. He is a board-certified dermatologist, of course, very, very well adept in clinical research in the area of dermatology, a key opinion leader in both atopic dermatitis and psoriasis, and founded Innovaderm back in the year 2000 as a clinical research site in Montreal, Canada. Next slide, please, Mikhail.
What I want to say is to thank you to all. Innovaderm now is the largest CRO, independent devoted to dermatology. Our company is different because our mission is to support the advancement of medical knowledge and development of new therapies. We do this in three ways. One, we manage studies for pharma and biotech companies. Two, and this is for-profit activities. Two, we use some of these profits to reinvest and do internal research that does not have a commercial goal. Currently, for example, we are doing a large study characterizing chronic hand eczema, the various subtypes. And three, we donate money to foster and fund academic research. With colleagues, I started a few years ago a nonprofit organization called the Montreal Dermatology Research Institute, and the Innovaderm Group has donated in the past 2 years $2.8 million to this organization to fund academic research. So this is possible because of your support, dermatologists in the room, and the pharma and biotech companies that have worked with us. So thank you very much. Have a good day and a good meeting.
This is me. We can skip my profile, please, Mikhail. And I'll have one slide on Innovaderm, and then we'll go right to our very distinguished speakers. So for those of you that don't know, Innovaderm is a full-service CRO dedicated to dermatology clinical research. We only work in dermatology. We don't work in any other therapeutic areas. The company was founded in the year 2000, like I said, by Dr. Bissonnette, and we are headquartered in Montreal, Canada. We started as a research site in the year 2000, and by 2012-13, we became a full-service CRO. So we've been a CRO now for about 10 years. We've managed over 150 trials as a CRO, have over 3,100 sites in our database of network sites across the globe, actually. We're now at over 400 employees worldwide with operations in both Canada, the US, and several countries in Europe as well. Next slide, please. Today I'd like to introduce our distinguished speakers. Dr. Amit Garg is a professor and founding chairman for the Department of Dermatology at the Zucker School of Medicine at Hofstra University. He's also professor in the Center for Health Innovation and Outcomes Research at the Feinstein Institutes for Medical Research. Dr. Garg was awarded the 2016 Thomas G. Pearson Educational Memorial Education Award by the American Academy of Dermatology. Welcome, Dr. Garg. Dr. Alexa Kimball is the CEO and president of Harvard Medical Faculty Physicians at Beth Israel Deaconess Medical Center. She's a professor of dermatology at the Harvard Medical School. In recognition of her research on physician workforce economics, quality of life, and outcomes, she was awarded the American Skin Association Research Award for Health Policy and Medical Education. She's also the recipient of Lifetime Achievement Award from the National Psoriasis Foundation and Outstanding Physician Clinician Award from the National Psoriasis Foundation. Last but not least, Dr. James Krueger is professor at the Rockefeller University and head of the Laboratory for Investigative Dermatology in New York. He also serves as physician and co-director of the Center for Clinical and Translational Medicine at the Rockefeller University Hospital. He's the Chief Executive Officer of Rockefeller University Hospital, and he's also the recipient of the American Academy of Dermatology Astellas Award and Society of Investigative Dermatology Farber Award. Welcome, Dr. Krueger.
Yeah, thank you. That it is really wonderful to see so much interest in this disease state from potential sponsors, 7 a.m. on a Saturday in New Orleans, no less. So congratulations to you all, and thank you for being here and engaging in this discussion. And thanks, of course, to Innovaderm for helping to put this together. My name's Amit. I'm a dermatologist. I practice in New York. Mostly see patients with inflammatory and autoimmune diseases, and certainly within that HS within that. And really one of my areas of research focus is in development of measures for clinical trials, and also with a focus in HS. And hopefully over the next few minutes we can understand why I think that's a very relevant thing at this day and time. Here are several relationships that I have. I guess they're relevant to this talk, since you're all here too. In my perspective, I really feel that the greatest threat to drug development in HS is this issue about high placebo response rates in HS trials. Or said another way, poor separation between the MOA and the placebo arm. And so what do we see? We've seen now, we've got some experience in several phase 2 and phase 3 trials in HS, and we're starting to see some pretty consistent patterns. We see that in many cases, the MOA response rate is actually pretty good. We'd like it to be better, but it's pretty good. But what we're also seeing is placebo response rates that are sometimes almost as good, but approximating it. And I think that's really been some of the concerns, with it. And we've always asked ourselves, is it something related to the disease? Could it be something in the protocol, in particular the measure? Or is it related to the people who are applying the measure? And we've thought about this quite a bit. I mean, I don't think it's the disease. I think we all appreciate that HS is a disease that has fluctuations. There's no doubt about that. Patients will tell you that the first time you meet them. But the fluctuations, in my experience and in general, are not great and worse, okay? It's not great and a lot better. So there's no reason, and I don't see in practice, 35% of my HS patients getting 50% better over a period of 12 weeks on no treatment. I feel like the trial is better at taking care of HS patients than I am if they can achieve, that result. I may have to refer all my patients to the trials for that reason. So I don't think it's the disease. Is it the measure? So at present, the current endpoint in trials is the high score. I think high score actually is a pretty, conceptually a valid sort of framework, right? It captures two of the most common and important lesions that we see in HS and that are important to patients, abscess to nodules. I think if there's a critique, it's been that it doesn't capture response of fistulas, and it's got this very stringent criteria that if you go up or increase by one abscess or fistula, that you become a non-responder, so that can be really tough. But the measure itself is not unreasonable. So then there's the raters, us. And I think that there's probably something in the rater piece that is accounting for some noise, at least, that we're seeing in the data. And why is that? Well, if you think about the disease itself and its presentation, it is quite heterogeneous. What does that mean, or at least what am I implying by that? Well, you've got your three main lesions, abscess, nodules, and fistula. And at least a couple of them can look very similar. I would argue, actually, they can all look similar in some ways, right? Even tunnels have little nodules at the end of them. Even abscesses can drain like tunnels can, right? So there's a lot of similarity in the three main lesions. And then you have within each of those three main lesions, right, other considerations. Is an abscess draining or not? Meaning, does it look like a draining tunnel more so than it does an abscess? Is a nodule inflammatory or not? That's important because we capture one and not the other. That's also important because, at least in the United States, we know that Black patients are disproportionately affected with HS, so darker skin tones, erythema, which indicates inflammation, can be a little bit harder to sort of pick up on. And then there are tunnels. Are they draining? Are they not? Do you push on it? How hard do you push, right? All of these are variables just in the 3 main lesions that we're supposed to keep track of. But then HS has a whole myriad of other spots, and they are relevant because in order to know not to record a papule, you have to be able to recognize a papule for a papule and not a nodule, okay? So in fact, these other lesions are important to distinguish from also the other main ones. So I don't know, there's like 7 or 8 things on there that can sometimes look similar, and so you can see why even experts have a hard time with accuracy. And we've shown this. We've gathered whatever it was, 12 experts and 24 patients, and we all got together in Denmark, and we said, let's just evaluate the same patients and see how well we agree. And lo and behold, few of us could agree on much of anything at all. And that was very disconcerting, but it told the story that there's probably a greater issue here. And we all know the truth that when you sign up a site for a trial, it's not even always the most experienced person doing the ratings. And that adds another layer of complexity and challenge to the ratings. Okay, so I think then there's good premise for suggesting that there's an issue with the raters, probably more than the measures. But having said that, how then can we overcome this problem? I think there are certainly technological things that we can think about. I'd love to hear everyone's perspective on it. But as it stands today, what are the things that we can do to try to overcome the inaccuracy that we have as human beings in trying to rate this disease? And that's where the measures then I think have some more relevance, right? The measures themselves are good, but what are they asking us to do within sort of the limitations that we have? So let me go through at least 3 measures that I think, obviously the current endpoint and 2 others that are in development to see if any of them may help overcome some of this. So the high score, most of you are probably familiar with, it's a count of inflammatory nodules and abscesses, and this is a dichotomous response defined as a 50% improvement relative to baseline, the AN count, and you can have no increase in abscess and fistula relative to the baseline count. And as I mentioned, the fistula here, well, technically it's included in the assessment, it's not included in the response, I guess, if you will. And I think that's an important limitation to consider. Patients will tell you that's probably one of the more, if not the most troublesome lesion. And I think even the FDA has at least sort of made this comment that we're all trying to figure out what does it really sort of mean, but at least essentially saying that yes, we think that tunnels are also important. Having said that, we're sticking with the current construct, at least for now. So that's sort of high score. IHS score 55 is a fairly new endpoint that has been developed and now validated initially as a continuous scale and now as a dichotomous one, because I think FDA sort of prefers these dichotomous endpoints. And the construct is actually very similar to high score, with one exception, is that it includes tunnels in the response. And in fact includes them, but also now heavily weights them to 4 times relative to, let's say, nodules. And I don't know, we can argue, is that arbitrary or is it actually right on point? Because patients will say that tunnels are the most problematic lesions. That's sort of arguably reasonable, but arguably also arbitrary. But it's been validated in several psychometric properties as a very good dichotomous response. I think the main limitation for me here is that it still does require, again, recognition and distinction of the 3 main lesions, and that's where that's the part that we're not that great at. So whether or not this overcomes some of the limitations we have as raters is unclear. A third instrument that has now been developed and validated, and my conflict of interest is that I've been involved in the development of this, is called the HS-IGA. But one of the construct is a global assessment, which we know the FDA likes for derm diseases. It's a 6-point scale, 0 to 5. But I think what's nice about this is that it's inclusive of all the 3 main lesions, and even the types within the lesions. So abscess doesn't matter whether it's draining or non-draining. The tunnel doesn't matter whether it's draining or non-draining. Nodule doesn't matter whether it's inflammatory or not. You just count them all. So the nice thing is you don't have to distinguish among them. And I think that's one of the, we think that's, may help overcome, one of the limitations that we have. But otherwise it's, similar to what you see with other global scales. So these are the 3 sort of measures. High score firmly remains the endpoint. I think the FDA sort of has stuck with this, even though they've been prompted to consider at least one other. So it's hard to know, if anything necessarily changes. And if it doesn't, we all just have to become much better at being able to recognize the 3 lesions, distinguish among them, distinguish within them, and accurately count them. So that's really an overview of what I think the main challenge is in ratings for investigators in HS trials. I'll give a quick mention to a couple of organizations who are very intensively working on these types of issues. The first is Cord Cousin Collaboration, C3, which is a group that is focused solely on developing core outcome sets for clinical trials related to dermatologic diseases. Again, with that appreciation that if we don't have measures, especially for some of these uncommon and rare diseases, that are robust enough, rigorously developed, validated, and robust enough, to use in trials and in a standard way across trials, then companies will have less interest in hundreds of millions of dollars of investment in studying their MOA for that disease. Really, the goal here is to support the care of patients for some tough diseases like HS, pyoderma gangrenosum, but also more common ones, vitiligo, eczema. All of these are groups that contribute to C3 for this work. And then the group that is, we're stuck here. Okay, it's stuck. But the group that focuses on this work for C3 in HS as a disease state is called Historic. And there are several measures that are being worked on that we hope will comprise a course that really will capture the interest of patients first and foremost. We think that their input in what they should be sort of measured on in trials and the response is relevant and important, but captures the perspective of industry partners, experts, methodologists in developing these measures that we hope will lead to more interest in drug development in this disease state because it's very much needed. Okay, thank you. And as was mentioned, I'm sorry I have to step out a bit early, so if there are any comments or questions related to this topic, I'd be happy to take them. Thanks.
Just raise your hand and I have a mic to if you have any questions at all for Dr. Garg.
I don't need a mic, it's Thank you so much for your talk, it was really interesting. I was wondering, what do you think is an expected or desired delta when you have a drug that works between placebo and active subjects because of the high placebo response?
Yeah, the desired delta is certainly a statistically significant one, first and foremost. I think the way that I could answer that is maybe more what is a desired response rate.
Yeah.
What I can tell you is that when you meet patients who achieve a high score of 50, they're very appreciative, but they tell you that their disease is still pretty terrible. And so I think what we certainly need is a higher bar, which means we also need more effective therapies, right? That do have to come sort of hand in hand. Now, high score, does that because there's high score 75 and 90, this construct that we've now, sort of started to talk about. And that gets patients to a higher bar still, ideally, with a delta. I think what I would say is I would love to see, a higher bar for response and more patients achieving it. But in terms of just, from a trial, sort of endpoint results that, a statistically significant delta would be great. I think placebo rates, ideally, if we're really doing this well, should be below 20%. I mean, 35, 40% is really high. I mean, we've seen up to 50%, I think, in trials that have failed, right? But I think, it's never going to be perfect unless we have, technological sort of solutions to help us as humans, do this better. But I think if we can get that to be under 20%, then even in the current landscape of efficacy, we should have more drugs that, are looking promising. Yeah.
May I add a question to it?
Yeah.
What do you think of the type 1 error when you have, let's say, a high score of 55 or, the economists comparables? Don't you feel like you lose a lot of information when you do a yes, no, when you have a...
Yeah. I would say for sure, HS is not a yes/no disease by any respect, and so but at the end of the day, the FDA is dichotomous. That's I think we've seen that over and over again. So, and I think IHS4 started as a continuous scale, maybe with the recognition that it's not as simple as yes/no or black or white. But I think, with the pressure of, having to sort of have a dichotomous endpoint, that's when they looked at IHS 455. All right. Hey, yeah.
Among all the endpoints, is there one which is less sensitive to placebo effect?
Is there, among the endpoints, is there one that is...
You have a list of endpoints.
Yeah.
Is there one which is less sensitive to the placebo effect?
I mean, what I would say is that the higher bar you have, so I think high score, if when you look at high score 75 and 90, the response rates obviously are lower, but usually the placebo rates are lower too, for a drug that has efficacy. So I think high score 75 and 90 can achieve that. Certainly IGA, at least in its psychometric assessments, have sort of achieved that. Because it too is a higher bar, it probably functions more like between a high score 75 and 90. So certainly with IGA, there tends to be a lower placebo response. Thank you, everyone.
Quick follow-up comment to the presentation. It was terrific, and I agree with almost all of it, but I would pose, I can't resist. I'll just say, I think we still have an open question about the underlying variability of the disease. We've been doing some work looking retrospectively at the Pioneer data set, which now is available on a patient level through Vivly. And I've been zeroing in on the high score placebo responders to see what other things are happening to them. And as a group, their CRP levels are dropping, their elevated platelet counts, their anemia in those patients who have those abnormalities which are related to their inflammatory load are also improving. So I do think that some segment of those placebo responders are indeed better. We may not be seeing them in clinic because they don't call us. And so I think all of these things are contributing to it, but I do think there is underlying variability to the disease that we haven't always appreciated, and those biomarkers are suggesting that that is happening to some extent. So I encourage, as we get broader data sets, that people start to look at that question. Right now, everyone actually has a whole cohort of placebo responders. What is happening in that group? And let's figure out a little bit more how that will help us get to the grading.
Could I ask just a quick question? Is that because the patients are being managed better during the course of the study, that they're paying attention to those lesions, or what is happening in that?
It may also be that the disease waxes and wanes. So if you look at atopic dermatitis studies, you look at psoriasis studies, when you had 50% improvements as your benchmarks, we always saw placebo response rates in the 30% as well. You can even see a placebo response rate in oral contraceptive acne studies where women are on OCPs or not. You would think most women would know, they would experience that, and yet those rates are high. So part of it is where the bar is, right? So what I would posit is that patients do fluctuate in and out of disease, and they vary 20 to 30% either direction. It's kind of a normal distribution. They don't vary 75% very often, and that's why you see those rates drop. So what we are experiencing is not very different from what we've seen in psoriasis and atopic derm development. And I will say, 20 years ago, dating myself, but 20 years ago there was a gargantuan debate about the PASI score and how terrible it was, that it didn't assess it, it wasn't reliable. And then as we got drugs where you got 90% improvement, no one ever talks about that anymore. So I hope we get there for HS. I think this problem will solve itself. I do think how we incorporate fistulas is really important, and we're going to need to continue to work on that. But my major lesson is stop worrying about the scale and just make me better drugs. What I'm going to do now is actually sort of step back. There's a lot of really hot, fabulous data that's being presented, some of it today. We all have to get over the late breakers, right? So I'm not going to touch on some of that, but just kind of give you where the clinician really is sitting with this to give you a sense of the unmet need and the choices that people are making and what we see and observe in the clinical arena that informs what the opportunities are. So I, of course, have a number of disclosures because I work with and design a lot of studies, and Tina Porter is my partner in crime and helps us all to keep up to date. I would say I pivoted to HS around 2009 with this aha moment. This was the index patient, not a very typical patient, but she had horrible disease. She had been to a surgeon. He had done gargantuan-wide excisions. They had dehisced, and they weren't healing. And he just randomly sent her to me. It was just one of those moments in fate. And around the same time, a colleague of mine at AbbVie actually called me and said, I'd really like to have some help in designing a scoring system so we can assess HS in clinical trials. And I just went, wow, everything that I've done in psoriasis, I now need to go do in HS. No one has done any of it. And that really was a pivot for me and this incredible journey that's come there. So same questions, to very different answers, and we've learned a lot. And Jim's going to talk about, I think, bench to bedside. I think we're learning a lot also the other direction as we see which therapies succeed in the clinic and which ones are failing as well. And I will say this patient, do I have it? Yeah, this is her now. I continue to follow her, actually. She really just comes back for social visits, and so it's really remarkable. And you know, she managed to get herself to social work school. She's gainfully employed. You know, a lot of really good things happened because we got her through this period that might not have happened otherwise. So, from a clinician standpoint, there's this many treatments, right? So it's been a little overwhelming in the clinic to figure out what to do. And a lot of sort of innovative therapy, and the patients are innovating in their own ways. When you survey them, they are using zinc and laundry detergent and all sorts of things in an attempt to try to get this under control. But we are really seeing some different trends in the clinic as we're starting to, I think, synthesize a much better algorithmic approach to, at least for people who don't do this all the time, give them a really good starting point. So we talked a little bit already about the scoring system, so I won't go over those, except to say that, you know, we are all going to have to figure out how to incorporate fistulas into how we think about assessing things, because they are such an important part of the disease. But for many of the patients in the clinical trials, they don't have any, and the ones who do may have on average 2, and so consequently it doesn't weight very well in the overall systems. And that's why you're seeing the IHS4 55 data look pretty darn similar to the HiSCR data, because while there is a fistula impact, it's really on the margins. So here's where we're really, I think, focusing from a clinical standpoint right now is, what are the phenotypes that we see in the clinic that help us figure out what therapies to apply? And there's a really important breaking point to this. So the most common early stage disease that we see, I would say, is the follicular phenotype. There are patients with what I call a comedonal phenotype that probably has more of a genetic basis and is quite rarer. And then there are some patients who just present with big abscesses, and that's kind of a different group. Any of those groups can then go into what I call an infiltrative invasive form of the disease, and that's the more classic early 2 and 3 that we usually see. So when you're still in earlier disease and don't have that pathogenic wounding kind of phenomenon going on, you can really use different therapies. And again, all of these can be used adjunctively pretty much with more advanced disease, but they'll have less impact. So hormonal treatment really has been very powerful, and spironolactone in our clinical setting for women with, again, early 1 disease can be a total game changer and the only thing they need to do. So the androgen rebalance is very powerful in reducing the inflammation at the level of the follicle, just like it is in acne. And it's a little surprising to me that it works as well as it does, given that these lesions are so much bigger and deeper and intense. But I have a number of patients on monotherapy. We haven't studied this as much as we would like. We've done some retrospective chart reviews. But it's very hard to, it was interesting, way back around 2012, I tried to do a study for Yaz and HS, which was absolutely the right question, but it was just not, you know, it was a time where there's a lot of controversy about that medication, and the patients were like, well, you could just put me on it. Why would I go into a study? So they were very non-compelled to enter, and I think that's where we would end up with spironolactone. You also see people doing finasteride, say proterone acetate, not available in the US, and then OCPs. And one of the most interesting things in the OCP world that I haven't seen much written about yet is there's a new one called SLIND, which is drospirenone known alone without estrogen. And that is really what spironolactone is. And it actually raises this interesting question for me, which is if, of course, they're metabolites of each other, if in fact drospirenone alone is a good contraceptive, then actually all these people on spironolactone probably couldn't get pregnant anyway. We counsel them all about it because it can be teratogenic. But in fact, it may never have been a problem, and I think that's why it's never really been a clinical problem. But I leave that out there as a thought. But we're seeing some nice progress, I would say, and that may help because some of the androgen IUDs and other things do seem to exacerbate HS, and so getting to a progesterone-only solution that can help HS would be important. And then there's some older data on metformin, there's people using semaglutide, other metabolic things with the same kind of concept. I would say I haven't found them clinically nearly as useful or as easy to use as spironolactone or OCPs. Of course, we also use antibiotics for the follicular phenotype. Usually on the right there, you'll see doxycycline, minnow, Clinda, etc. We use amoxicillin and Augmentin for flares quite frequently. Retinoids are in the literature, and I would say particularly for male patients, right? The real issue, Accutane isotretinoin is not particularly effective. You really have to go with acitretin. And for women of childbearing potential, which is a lot of our patients, that's going to not be an option at all. And so I do use this as an adjunct primarily in men who are on immunomodulating anti-inflammatory therapy where drying things up a little bit may be helpful. And again, for those where follicular differentiation matters, that can sometimes help too. Dapsone is a nice add for people also on anything that is chronically suppressive from an immune system standpoint because it prevents some opportunistic infections. Again, I'd say modestly effective, but sometimes a nice choice for patients who really are nervous about a classic antibiotic and resistance for a long period of time there. So you can see, like, we're mixing and matching a big soup of stuff. There is kind of a natural algorithm, male, female, and then what do you do for the early stage disease? And so the options become pretty straightforward, at least in our clinical setting. But then, as I mentioned, people start to move into a more invasive form of this. And so an infiltrating form where we have to consider biologics or JAK inhibitors or some other approach. Those are typically patients who are early 2 or 3, high inflammatory burden, or higher. They start to have scarring, flares, and aren't responding. I mean, oral contraceptives are not going to touch this disease, right, at this point in the game. And so you need to really think much more aggressively. And you need to do it quickly because once people start progressing, it's very hard to keep ahead of that progression. And I think in the last 5 years, I've gained a much stronger appreciation that there are a cohort of patients who rapidly escalate over a 1 to 2 year period of time. We could see it in the UNITE registry data, and I was surprised where I saw it there. I said, well, I didn't see this clinically, but now I do. That in fact, and you're just chasing them. Like every time you see them, you're bumping up the therapy and they're still getting worse. And then you're going after them. And those are the patients where you've got to get ahead of the game. So adalimumab is the only FDA-approved medication for moderate severe HS at this time. That was approved in 2015. So a little bit of a journey gap between the innovation we're seeing now. Most, at least in my experience, you can get this very easily for most patients without step therapy or anything because it's approved. I will say we are also getting 80 milligrams a week quite easily now, which is a little bit surprising to me, but now that there's an 80 milligram syringe, that's actually not very hard to do, and that can be very helpful. Infliximab is still a very good standard for severe or escalating patients, especially because you have such dose flexibility in terms of trying to get ahead of it. And then, as we've seen in the last few months, and as we'll continue to see through this meeting, IL-17 is now a validated pathway, and I think opens up for us to figure out what the right dosing and strategies are for these patients, but it's clear that this works. JAK inhibitors, again, multiple studies now really validating JAK1 in particular as a validated pathway in the disease. And then the IL-23 inhibitors have had their studies essentially concluded or terminated for lack of efficacy. So, whereas we may have thought that 23 being so important in the skin was helpful, it appears this IL-17 pathway is IL-23 independent. So we are going to have a lot more to learn to get there. But exciting, right? So we had sort of phase 1 TNF, phase 2 IL-17, phase 3 on the horizon is JAK, and then a number of other new modalities, and I think Jim will talk about some of those as well. And we've talked a little bit about the data, so I'm not going to spend much time on this, but to just show, again, we're looking at deltas in the 25 to 30% range. Infliximab, I thought this graphic was very useful in demonstrating the doses that we need are high to get control. This shows you what happens to the dosing algorithms for the patients, and this and also Dr. Cohen's data really suggest starting 10 mg per kg Q4 at the very beginning, so very aggressive treatment regimens. Secukinumab data was just published in February, and you can see at week 16 there that you have a similar type of delta in the 20-point range. And I think as we're seeing with almost all the biologic therapy, persistent improvement as people carry forward. Open label extensions are of course hard to interpret, and as observed data, but I would say also clinically, this is a disease where you need to treat aggressively to capture it, and then you do see continued improvement with good therapy in a lot of the patients, and not that same drop-off that you sometimes see in psoriasis. I'll be presenting the phase 3 bimekimab data later on this afternoon, but just the teaser that here's phase 2. So bigger study coming, that's all I can say. And then this was the first set of patients case report we did using tofacitinib in HS. These were incredibly sick patients, essentially bedbound or wheelchair bound because of their immobility, because of the pain and ulceration. We treated them with a whole bunch of other things, but the secret sauce, because we treated them with many, many things, turned out to be JAK inhibition, and that's really what got us to improvement with these patients and started me on this journey of trying to figure out how to use it. And again, a number of studies, this is just one that was published earlier this year of porphyritinib showing high score deltas that you can see depending on the dose, again, in kind of that 20 to 30 point range. And I will say, I'm calling this the HS wobble, which is this funny thing that sometimes happens, and it's happened more than once in HS, it doesn't happen in other disease states really, that the second to last data point is cleaner than the last data point for some weird reason. So there do seem to be a few study characteristics that lend to that, so we're doing some work to try to tease that out. And then last but not least, there's the HS sort of chat listserv, and people are trying all sorts of stuff. And one of the sort of favorites that are kind of in the background is ertapenem, which is both expensive, ID hates us to use, and requires daily IVs, but people do use it for rescue therapy. And that's inspired me to think a little more creatively about sub-Q and IM injections for patients who need antibiotic therapy for a period of time. Things like IM ceftriaxone, which is a daily antibiotic, are actually in Europe given as sub-Q. And that could open up some ability to do some other things. We do a lot of work with prednisone and augmentin as well. We actually just completed a study. We had over 150 patients call in a flared state. And again, that's that's a lot of patients on a reasonably sized cohort. But this treatment regimen is a great way to manage them. And we only had one patient go to the emergency department, and she went before calling us. So this is a great strategy to keep people out of the ED, and this type of stuff I think will help our clinicians. We've also been working on ways to manage fistulas in a less surgically invasive way by putting in hypertonic saline into the tunnels. And you can see that it usually requires a few series of treatments, but it is completely non-morbid other than some stinging because you're going in the tunnel that already exists. You don't even have to break the skin to get there. And so I think, so inexpensive, low morbidity, and hopefully maybe helpful in some patients. So I always say to patients as I conclude, this is a journey. It's going to take me a year to kind of get this to the right framework of hopefully a sort of remission, but it's not going to go like this. It's going to go like this. HS has lots of bumps in the road, and my goal is to get you here where we're just kind of in a wave. But you know, we're going to work together to get to that place, and so it's very important to set that expectation and tell people that they're going to flare and what to do if they flare, so they don't go to the emergency department or suffer at home. So in general, it is just an amazing time for our patients. They're excited about it. We still have a ways to go, but the progress is enormous, and excited to be on the journey with all of you to get us to this next level. So thank you very much.
Any questions for Dr. Kimball?
Thank you for an informative presentation. So my question actually is, what combination of therapies do you frequently use?
So I would say in the earlier stage disease, fine to use, for example, hormonal and antibiotics. So that's kind of my entry point. You know, so I'll start with one or both of those as we go through. As you get to more advanced disease, not uncommon if I'm using a biologic to include hormonal therapy for women, because it's easy to do. They often tell me they flare around their period, so can't hurt to kind of get that into the mix a little bit for them. And then, you know, I will either combine a retinoid with a biologic, for example. I will, it's hard to get a biologic and a biologic, although for some patients that probably would be beneficial. But I'll also combine a biologic with the lower dose of a JAK. And that's been another good strategy. If I just, so I try to dose escalate my biologic first to the max that I can, and then add in a JAK inhibitor and or an antibiotic. So it's kind of building through it.
Any other questions?
Why you don't start earlier with an aggressive treatment? So starting the biologic sooner than later, before the disease would make it out of control.
I mean, I think it's a great point, and there's a couple of reasons why. One is most patients who have the mild disease aren't kind of mentally quite ready to make that jump. The second I see them start to have tunneling, though, I give them the talk and say, we got to go after this now. But I do have decent success. I have good success with the earlier follicular disease with these other modalities. So unless I see them in that progressive state, I have people in remission on like 75 milligrams of spironolactone. Like, it's hard to beat that from a safety risk standpoint. But as I said, once they convert to infiltrating, you have to be more aggressive. Okay, thank you.
Thank you, Dr. Kimball.
Is the advancement on? So these are my disclosures. There are not so many around HS, but virtually any company that's pursuing HS has also pursued psoriasis at some point, and they are widely conflicted. All right, so you know, it's a really interesting time for HS. And Alexa and I have talked, and you know, sort of what's happened in the last year has given us hope that this disease is seeing a renaissance of translational science. It's going to help us get to where maybe psoriasis is. We're very lucky. So psoriasis is the best treated inflammatory disease in humans, period. And so with single drugs, we can expect 90% success, right? And we've gotten there by a 25-year period of this approach of translational science where we try to understand the underlying cellular molecular disease characteristics in patients, and from that we pick therapeutics that may or may not work, and we try to study those by biopsies and molecular approaches as we go. Sometimes we're right and sometimes we're wrong, but we learn from successes and we learn from failures. And I think we're at the stage of HS that here we have a disease that's way more complicated than psoriasis. We've already talked about early stage 1 versus early stage 2 and 3. And you know, a lot of the current thinking about what's going on in HS stems from you know, disease pathological characterization that's been published almost in the last decade, right? And And it's really from this study that that you get this early view of pathogenesis that's really folliculocentric in terms of saying there's a hair follicle, perhaps it gets plugged, there's inflammation that goes on which causes it to swell. At some point the wall ruptures, that leads to inflammatory products that may bring innate immune cells into the picture. And if you do this with enough follicles close enough together, you might get a fistula that's created just by the fusion of different tunnel areas that have broken down. Now, this model doesn't have any part of it has T cells in it, and the argument really is based more on innate inflammation. We know from what Alexa's already shown you with IL-17s being effective that these IL-17s originate from T cells. So T cells are in fact part of the picture. Now, there was a paper published 2 or 3 months ago by Shaw in the British Journal of Dermatology, which began to look at inflammatory characteristics that included early stage 1 patients. And it's also not focused on follicles, but it's focused on the interfollicular epidermis that is associated with early HS lesions. And what they found, and stage 1 aren't here, but they showed cellular infiltrates of T cells and other cells that were heavily increased in or near the epidermis, the earliest lesions. But then also the idea that even as one progresses, there's an increased expression of cytokines by epidermal keratinocytes that relate to molecules that include IL-17 and IL-17 pathway molecules, as well as innate and adaptive inflammatory pathways that go up. And they tested some JAK inhibitors for activity in cultured cells that came from HS patients and saw that cytokines could be suppressed. And I think what this leads to is maybe a slight revision of the early HS model, where there is inflammation outside of nodules, and it may be modulatable through surface suppression, perhaps the topical drugs. And so this was a study that was sponsored by Insight, or at least had testing of Insight JAK inhibitors in it that showed suppression. So you could imagine that early disease might also be managed by a topical JAK if it doesn't go deep. And very early, it may not go deep. So the idea that this is folliculocentric, I think, has to be put held with a grain of thought. Now, my job has been, for the last 4 or 5 years, has been to try to take on the challenge of HS from the standpoint of tissue and immune pathology. And I will tell you that there are many different elements that you see on this slide that are both from background genetics and the dysbiosis of the microbiome that go through T cell, D cell, T cell, TC, and keratinocyte interactions that lead to a neutrophilic inflammation that we've been talking about. I'm not going to talk so much about it, but there's also a very, very strong B cell and antibody component in the disease. So this is a mixed disease from the get-go that's kind of akin to lupus or rheumatoid arthritis, and not like psoriasis or atopic dermatitis, which are really T cell driven diseases of the skin almost completely. And what I want to acknowledge is this work was really started when John Frew, who's an Australian dermatologist with an interest in HS, came to Rockefeller as a clinical scholar and started working with Christina Navrazina, who finished her PhD dissecting elements of HS that I'm going to show you, and will be a dermatologist working at Mount Sinai in the future. Now, our approach has been to try to look at lesions, ultrasound lesions, identify tunnels, do biopsies, and study this by broad omics. And just to give you a picture, so a tunnel is something you can't necessarily see from the surface. Maybe you can infer it by the swelling. You put a probe into an ostia and extend it, or as Alexa does, put hypertonic saline in and get a sense of the direction of a tunnel. But the fact is, you really can't see it because it's a deep dermal structure. And so we've been able to take complex lesions And we're interested not in the nodule, but in perilesional skin in the most part, where we're not seeing pathology at the surface, and then we ultrasound and we see these parallel echogenic bands, which are echogenic like the surface epidermis that you see here. And so if we do a biopsy of this ultrasound picture in different patients, what you see all underneath these parallel echogenic bands are epithelia that look a lot like the surface epithelia. So you've really got two disease components that I would argue, are reflected in different disease measures clinically. The first is this surface psoriasiform conversion. Frankly, if you looked at this alone, you'd say, oh, this could be psoriasis. And then there's this deep dermal epithelial structure, which is surrounded by massive numbers of neutrophils and other inflammatory cells, and many perivascular inflammatory cells above and below these structures. And where is inflammation maximally happening in tissue? Well, we measure that with Doppler ultrasound, and so it's, which I'll show you is around this, you've got this huge amount of blood flow, which is indicative of inflammation. So these are inflammatory structures. Now, at a molecular level, we do things like RNA-seq and compare this to normal skin. And what you see here are volcanoplots that are of perilesional skin, which is 2 centimeters away from a nodule or an abscess, and lesional skin, which is a biopsy right at the edge of one of these nodules, inflammatory nodules or abscesses. And here's non-lesional skin that's 2 centimeters away. There are about 3,000 dysregulated genes in HS, and as an idea of the magnitude, this is about the same number you see in psoriasis or you see in atopic dermatitis. And you can point to individual elements here, and there are things that are shared with psoriasis, such as S100 proteins, and also chemokines like CXCL8 and products of neutrophils. So there's a neutrophil signal that's in this. And so if one takes, the sum of these 3,000 products, we put them into objective analysis pathway tools, what you get out is at the very top of this pathway is neutrophil degranulation, and then you see other things related to immunity, but you see T cell signals and other signals that are in it. And there's full disclosure, I'm taking the B cell signature out of it, but if we had it here, there would be many B cell signals also. But I've done that just to make me think about the T cell adaptive immunity side to a more clear extent. Now, we also think within HS, there are likely endotypes in which some patients are highly inflammatory, and other patients that may look the same clinically are less inflammatory in their tissues. And one of the things we've used to divide this is the molecule methylcalin-2, which you see here. And methylcalin-2 high patients tend to have very high expression, shown by red, of this array of inflammatory products, which include not only CXCL chemokines that would attract neutrophils, but also neutrophil products themselves, as well as granulocyte colony-stimulating factor, which of course may drive granulocyte production in the bone marrow. So this is a very strong inflammatory phenotype here, and this is, we chose methylcalin-2 because it's used in IBD as a fecal measure of inflammation that's driven. Now, many of these elements that we're seeing start to suggest overlaps with not only psoriasis, but also we know that they are IL-17 induced in tissues, and not surprisingly, we can measure IL-17 levels at practically the same as in psoriasis. So A and F are T cell, IL-17C is an epithelial keratinocyte-derived IL-17. And note that IL-17F is even higher in HS than it is in psoriasis, and that may be relevant to the presentation Alexis is going to give this afternoon with bimekizumab. And then there are IL-36 isoforms that are also elevated in HS, just as they are. And so, I started with trying to apply the psoriasis model to HS, where we've got T cells that make IL-17 and changing tissue phenotypes in large part through inductive effects in epithelial cells. And I think this much of the model has strong parallels with HS. But this part, where IL-23 is the factor that drives pathogenicity of type 17 T cells in psoriasis appears not to be the case. And while type 17 T cells in psoriasis, lots of IL-23 receptors that create that pathogenic conversion, you see the type 17 T cells in HS really lack IL-23 receptors, so you wouldn't expect that IL-23 blockade would necessarily have any effect. Now, we think a large part of the neutrophilic drainage in HS is mediated through tunnels. And so we have the tunnels as an epithelial structure that has undergone psoriasiform conversion, similar to the surface epithelium. And here is a tunnel with a lumen, and you see neutrophils staying in neutrophil elastase. You see these long strands, which are neutrophil nets. And so these are coming from neutrophil activation right at the edge of the tunnel, and migration of neutrophils through the wall of the tunnel into the lumen, creating what we think is this end phenotype of pus draining out of the surface osteum, which is connected directly to an underlying tunnel. And this tunnel may go on for centimeters here, creating a very large epithelial surface, which can mediate the entry of many, many neutrophils into the tunnel, creating this kind of drainage. And so, as you think about therapeutic targets, I put up, this map of thinking about the end product being neutrophils, and how do you get there? Well, you get there by either innate pathways that can be TNF driven, down classic sepsis cascade can be amplified by molecules like IL-36, and of course, these act on neutrophils through CXCR1 and 2 receptors. There's a presentation later this morning about blocking receptors to prevent neutrophil migration, and come about from adaptive immunity through the psoriasis pathway that I showed you, where IL-17A and F produced by T cells act on keratinocytes. You get amplification of this through IL-17CR36, and you get CXCL chemokines out, and there's a synergy that goes on between innate cytokines and IL-17A and F that really drives this production to extremely high levels. So both of these could be going on in parallel, and to think about whether how many of these things here are targetable and what makes the most sense to target to get the biggest effect. And I think not surprisingly, if we do unsupervised clustering of data, we find that patients who have tunnels have more inflammatory skin phenotypes by molecular profiling. And again, what's here is largely neutrophil centered in terms of inflammatory markers. The other thing I want to say is, IL-36 remains an interesting target across inflammatory skin diseases, and I think that includes HS. And it's been unrecognized for a long time just how inflammatory IL-36 can be. So this is an in vitro experiment where we've added either IL-17A or IL-36 to keratinocytes, and you can see each modulates more than 1,000 genes. In keratinocytes, there's an overlap, so there's similarity, and up here you can see heat maps of things that are reduced singly. But the really interesting thing is you put the two together. You get now many thousands of genes that become activated. You can see the degree of activation by the color of red. And so you have to think about cytokine interactions in creating these highly inflammatory phenotypes that may go on the skin. Of course, this argues potentially for combination therapy downstream to hit the critical nodes that lead to synergy, such as these. And here's the top 50 genes that are upregulated by this IL-36 stimulation that I've shown, and it includes many of the molecules that we've been talking about as characterizing HS. So this is our working model of HS. An epithelial reaction may involve follicular occlusion, leading to nodules that could be follicular in origin, or frankly could be caused by another process. Early stage 2 and 3 with progressive growth of tunnels and with inflammation associated with the tunnels. And another map, just to allow you to think about this. So we know that psoriasis is an inflammatory disease in the systemic circulation. And that's because there are about 50 different inflammatory proteins that can be measured in serum compared to normal controls. But from that standpoint, we've also compared HS to normal controls in psoriasis, and the number of inflammatory molecules is much, much larger, and includes some unique targets that are over here on the psoriasis side, on STAT10, for example, that might have consequences in JAK signaling. So, here's a map that allows you to pick out a highly expressed inflammatory cytokines. We know that TNF is a validated target in this disease, but you see what the levels are. It's surprising to me that even 80 milligrams of adalimumab may be capable of neutralizing all the TNF that's here. And that may argue more for why infliximab is so much better, because you're adding 10 times more neutralizing molecule. Now, therapeutic targets, we're at the stage, we're a very exciting stage. We've got some validated like IL-17 and TNF. We've got some things that have failed recently. And I think those failures are really important to help us frame the pathways. There are things that I think are less clear or potential, and in part, this is one measure of the clinical disease improvement, such as a tunnel might improve a bit, but the study was designed around a high scar, so you're looking at nodules, and nodules don't improve as much, and that needs to be sorted out. There's speculative targets. You're going to hear about one of them today, the chemokine receptors that mediate signaling of CXCL chemokines that cause neutrophils to appear at inflammatory sites. Signal transduction, certainly JAK1, maybe also JAK3, the IL-1 recessory protein, which mediates IL-1 and IL-36 signal, and these possibly downstream of the different kinds of leukocytes that I would say should include B cells, at least as they try to figure out what they're doing. And many other possible targets, including elements of the microbiome. So the field is still wide open. But I think we have to ground what we're doing with links to disease pathology, and also if we're studying targeted antagonists, we need biomarkers of effects so that we really know what's happening and what's not happening. So here's the crystal ball for the future. And you'll see that it's pretty cloudy. At least to me. All right, so listen, happy to take questions.
Any questions for Dr. Krueger?
Do you think that the two endotypes you were showing with the LCN2, for instance, can that be used for treatment to sort of predict to respond to IL-17?
Well, that's a hypothesis that's on the table. It needs to be tested. But I think generally, and Alexei, please wait a minute, I think generally as you get to higher levels of inflammation in HS, the ability to show a reduction in inflammation is higher with that, and you may have more success in more inflammatory phenotypes and anti-inflammatory treatments. Beyond, assuming you don't get so far out in inflammation that it's an impossible pass with mid-stage disease. And I think, the association of vital count too high with higher IL-17 expression certainly leads you to a hypothesis that maybe these would be more modulated with an anti-IL-17. So that's a prospective model, and it needs to be tested. And I think there actually are some existing data around which might actually allow that to be retrospectively tested from phase 3 trials that have happened. Right?
Yeah, I would say clinically and kind of as you look through the trial data, the patients who are less severe tend to be more responsive to therapy in the biologic realm. They also tend to have more placebo variation. The patients who are more severe tend to be less responsive but have less placebo variation. So that kind of makes sense. The delta is about the same. But there is sort of this sweet spot, middle spot that we haven't really evaluated. So the really severe patients, to your point, Jim, like you could throw a truck at them and it's hard to see a lot of improvement. So and then the early ones sometimes are a little disappointing in their response to anti-inflammation. So it's right in that early 2 range where biologics are the way to go.
Yeah, and I would say tunnels are permanent structures. And I think as this disease moves on, you can have a burnt out inflammatory stage, which is highly scarred, has many tunnel ostia, but but it's no longer actively inflammatory underneath. And now your problem is lack of mobility and maybe pain, but it's no longer approachable by the anti-inflammatory therapy.
You get Hurley 3 mild disease, right? So it's not intuitive. Hurley is a staging, much more like oncology, right? So this tells you how bad the patient has been, right? So you treat a patient with breast cancer who is stage 3, what are they today? Like, you couldn't do that in psoriasis because you eliminate the disease. But now you can have a Hurley 1 who is severe, and you can have a Hurley 3 who's actually mild. So people mix and match those.
The synergy that you have seen between IL-36 gamma and IL-17, are you seeing also this synergy just with this combination or IL-36?
No. So the way the IL-17 responsive genes promoters work is there's one CEBP transcription factor element that's a primary thing that's activated by IL-17, a receptor, and there's a second NF-kappa B binding site in the same promoter. And so if you get both of these promoter elements activated, you get the synergy. So anything that's an NF-kappa B activator, be it TNF, IL-1, IL-36, drives synergy. But the idea is you've got these two elements, and you may need to think about how to modulate both sides. Although just getting good inhibition of one side, may knock things down by enough, 10 times relative to the synergy that it looks like that one thing is active. It's not quite enough, right? You may need to go to both sides.
So you showed on your slides the whole potentially to target regulatory T cells, but you probably won't do this just targeting these. So what else could you do? I think you are thinking a little bit about combinations and such.
Well, all right. So I think the regulatory T cell problem is potentially huge in inflammatory skin diseases, includes not only HS, psoriasis, and so on. And so if you look at transcriptional profiles of Tregs across these diseases, you see that Tregs are largely dysfunctional because instead of making just IL-10, which is the anti-inflammatory cytokine, they start making pro-inflammatory cytokines that include IL-17. So they, IL-17 producing cells are plastic, and it's made in these diseases, the Tregs are co-opted into being pathogenic cells. So we need something on the Treg front that's not only going to potentially expand them, but improve their function. And I don't know what that's going to be. It may be that some of the background anti-inflammatories are improving Treg function as we go, but we got to look at it, right? It's not something that that can be intuited outside of actual studies, right? So work like single cell work today or flow cytometry is needed to really understand what's happening to Tregs, particularly in a therapeutic context, and that hasn't been done at all. Okay.